Good morning, and thank you for joining us. We welcome you to the third of the CASIS seminar series on tissue engineering and regenerative medicine on board the International Space Station, hosted by the Center for the Advancement of Science in Space, CASIS, manager of the ISS National Lab. Good morning. This seminar is being recorded and will be available at a future date on the CASIS website. My name is Liz Warren. I'm a Senior Program Director for CASIS, and I am honored to be moderating this final session, which will focus on stem cells. During this seminar series, we are reviewing research activities from the past couple of years and discussing the future of tissue engineering and regenerative medicine research. This series also discusses specific areas of research and new capabilities that enable life sciences research and in-space production on the space station. Behind the scenes here is a stellar team at CASIS who have worked really hard to bring this seminar to you today, and I want to be sure that I take a moment to thank them. Normally around this time, we would be gathered in person at the ISS R&D conference, but due to the COVID pandemic, we, like many others, elected to shift to an online platform. I encourage you to visit our website at issnationallab.org to learn more about virtual ISS R&D opportunities so that you can engage with the community. Today's topic is stem cells, a key component of tissue engineering and regenerative medicine. As you will hear in the following presentations, a number of studies on the ground and in space have indicated that microgravity may be a solution for many of the challenges of terrestrial clinical stem cell biology. I have the privilege today of briefly introducing you to our speakers, who will each spend 20 minutes presenting their topic. We will then host a 20-minute panel session that is open for your questions, and we look forward to your participation. So please feel free to enter your questions in the questions window to the right. Our moderators will collect the questions and will pose them to our speakers during the panel session after the presentations. I also encourage you to visit our website to learn and read more about the complete biographies of our esteemed speakers, as I have limited time to introduce them today. So we will start off with our first speaker, Dr. Abba Zubair. Dr. Zubair currently is the Medical Director of Transfusion Medicine and Stem Cell Therapy at Mayo Clinic, Florida. Dr. Zubair is also the Associate Dean of the Mayo Clinic School of Health Sciences. Dr. Zubair just published the results of a spaceflight experiment in June, and today he will discuss the expansion of human mesenchymal stem cells for the benefit of humankind on Earth and beyond. Dr. Zubair.
Thank you so much for the nice introduction. It's really a pleasure to be given this opportunity to talk to this audience. So as was introduced, I'm going to talk about projects that went up to the International Space Station. With the project's primary objective is to evaluate the feasibility of expanding human mesenchymal stem cell. These are primary cultures, not cell line, but freshly derived human mesenchymal stem cell expanded at the International Space Station with the intention of bringing them back to us for human application. We have several clinical trials going on that involve mesenchymal stem cell, and we believe that would form a platform of future human application of space-grown cells. So why taking stem cell to space? And why do we utilize microgravity? I think the short answer lies on this principle. Obviously, we're talking about growth or expansion. So at the heart of it is cell division. And when stem cell in our body divide, they undergo what we call asymmetric self-renewal. basically one stem cell would divide into two daughter cells. One daughter cell would retain all the characteristics of the parent and stay as stem cell, while the other continue to proliferate and give and differentiate and give rise to matured cells. In this process, stem cells are able to maintain their number constantly. So it's much harder to really expand stem cell, even though we found tricks in the lab where we can expand them. But inherently, they are designed to keep their numbers so that at any given time, the proportion of stem cell is about the same, which is a big challenge if you are planning to expand and grow cells for a number of patients, not just one patient, but giving it to multiple patients. So you need to grow it, and that's the challenge. If you normally cells that are not stem cell undergo symmetric differentiation. So basically the cells divide and the two daughter cells do not have the characteristic of the stem cell. And these daughter cells further differentiate to give different types of cells. Obviously, with induced pluripotent stem cell technology, you can reprogram the daughter cells to be back to having stem cell characteristics. So the main question is whether we can be able to induce symmetrical cell expansion or stem cell expansion in under microgravity condition. We know gravity play a role in virtually all our physiology. I tell my students that if human race evolved on Jupiter or Saturn, we wouldn't look like the way we look now. We would be a lot more like a lizard or something that is more flat, because the gravity there is much, much more than on Earth. So we know that gravity play a role in cell division, in chromosomal segregation. We also know a gravity impact on gene expression as well. So the question is, if you take it out, would you be able to reprogram these cells safely to expand without, making them unsafe? So we chose mesenchymal stromal cells. Some people call it stromal cells, some call them stem cells. So MSCs are actually maybe the most common or second most common cells that are used clinically in regenerative medicine. They can be derived from bone marrow or cord blood, cord tissue, or adipose site, so from fat. So the access is quite reasonable. They are defined by some markers, CD73, CD90, and 105, or absence of some markers, which are mainly differentiating them from hematopoietic cells. All these markers are not unique to the MSCs, but a combination of them kind of define the MSC. The International Society for Cellular Therapy also add to this definition that MSCs should have the potential to differentiate into bone, cartilage, or fat. So osteocyte, chondrocyte, and adipocyte is one of the key characteristics of MSCs. So I started by mentioning that obviously stem cells are used clinically, and so they are highly needed. Patients can donate their own stem cell, but not every patient are able to do that. Some of the patients, for example, that have leukemia or lymphoma, their marrow may not be adequate for even donating the cells. The stem cells themselves are damaged. So there is definitely need to have a donor or a healthy donor to donate. But there aren't many healthy donors that are available to donate. And then even if they do, it's harder to expand the cells to treat more than one donor. And we know based on, let's say, the bone marrow transplant activities that we do, we need about 200 to 500 million stem cells to achieve therapeutic efficacy. Again, in the setting of, let's say, leukemia, lymphoma. Of course, some application where you are putting the cells locally, you may even need just 5 million or 10 million or less. But regardless, you need a lot of cells to treat a lot of people, especially if you have one donor donating to multiple recipients. So we don't have effective method to really expand the cells. We do have some method, but it takes time. At a minimum, you need about a month to grow a reasonable number of cells to treat a few patients. And even if you need up to like 200, 500, you may need a lot more, or you need a bigger facility, a lot more resources. So scaling up is always a challenge, and you can see that the space are limited. You need more incubators. We are using multi-layer flask. All of that, and there's even automated bioreactors. All of that is quite pricey and difficult and takes time. So we asked the question, can stem cell expand in microgravity safely? What is the feasibility of doing that, especially if you want to generate clinical grade cells? So yeah, so we want to evaluate the safety and efficacy of using these cells for clinical application. Obviously, the journey to International Space Station was quite exciting. It takes time to get on the bus. What I mean by the bus, the only rocket at the time when we went is SpaceX that could take research payload. So you need to be on the queue, and for us, it takes almost 2 years to get on the rocket. This is Peggy Whitson. I tell people she's the most expensive lab technologist I've ever had. Obviously, being the astronaut that worked on our project, it's really a pleasure and a great opportunity to have an astronaut work on our project. And luckily, she also have a biology background, so that really helps. So what you are seeing is the container, the cassette that we use, BioCell, in collaboration with our implementation partner, which is the BioCell from University of Colorado. And that's how we were able to transport the cells in a PHAB and then to the International Space Station. It's quite challenging. We had initially thought we can do a lot more at the station, evaluating the cells, but reality set in, so we had to scale down a lot of things. In part, you got an astronaut who may have up to 20 projects working on yours, so you are just one of the many who is not really a basic researcher, so you have to really scale down. And then the setup also takes quite some time, like the use of microscope and stuff like that, or media exchange. We had to even do away with that in space. But anyway, we took 3 different types of cells: hematopoietic stem cell, leukemia cell line, and then the mesenchymal stem cell. So this presentation really focused on the MSCs, but we also look at other cells. We were able to get real-time imaging as the cells grow, and then we took the same pictures of the control cells in our lab. So you can obviously see it wasn't that clear, but at least for the leukemia cell line, the ones that are growing at the International Space Stations form clusters, while the ones that are growing in our lab didn't. And you can see also, it looks like the MSC are kind of more enlarged compared to the ones that are in our lab. So, but it gave us some clue. But that's all we could do at the International Space Station. Beyond imaging, we just preserve the cells in multiple ways, some in RNA protect, some because we want to further grow the cells when they come back to our lab. So we use cryoprotectant DMSO, dimethyl sulfoxide, and some we just fix the cells in formalin. So we had multiple ways of preserving the cells so that we can do further analysis when they return. So they return via this return capsule. Actually, this is the exact capsule containing our product. It landed somewhere in the Pacific Ocean. You get fish out, I mean, obviously using GPS. And then actually our samples were mailed by FedEx. So from, International Space Station down to us, and then the FedEx pick it up and deliver the cells to us here in Mayo Clinic, Florida. So it's quite remarkable that you can get your cells back by FedEx and safely. So when the cells get back to our hands, there's so much excitement, and the people were asking, what is the result? I tell people, well, wait a minute, that's actually the real experiment starts when we get the cells, because we couldn't do much at the International Space Station. So, and we were very careful not to throw out all our cells all at the same time, because these are precious samples, so we were very careful. But we did cell characterization, looking at cell count, viability. We did cell cycle analysis, gene expression profiling, growth factors, and then also look at differentiation. I may not be able to present all the data on this, but it was quite remarkable, and more to come, and we're still doing the analysis. The very important mission for the objective for this mission is really safety of the cells. So we wanted to see whether there's any radiation damage, so like DNA damage of the cells. Also, we wanted to see if they transform. And then we want to see whether they retain their regenerative capacity and induce tissue repair. So that is still ongoing, and I wouldn't be able to touch on the efficacy beyond the immunosuppressive activities. So quickly, these are the characteristics of the cells, before flight. You can see the cell identity that expressing those three markers is quite high, 89% viability, 96. And then these are the cells that we started with. Obviously, they are sterile. The Gram-stain and cultures were negative. We culture the cells for 1 week and for 2 weeks. So the data on the 1 week cells, you can see the identity is comparable between ground, which is a control, and the cells that were grown at ISS. You also can see the viability is reasonably comparable, even though the viability is a little bit lower for the ISS compared to the one in our lab. We are suspicious about maybe this is just due to the way this has been cryopreserved. We couldn't use control rate freezing, but I mean, obviously it has traveled this far, so maybe the viability is a little bit less. The cell growth is also actually we see a little bit more cells on average, but overall is within the error margin. We think the growth rate is comparable, and the same for the 2 weeks culture as well. The viability is a little bit worse, especially the ones at ISS. But again, we think that could be also related with the cryopreservation method. All of this is sterile, and that's very remarkable. We're worried about microbial contamination during processing or transportation, but all look good. We also tested their ability to proliferate, so we saw the cells and further culture them. The figure A is looking at real-time imaging using IncuCyte as the cells proliferate. And then on the figure V is showing the population doubling time. The summary of it is that we didn't see any of the significant difference between control versus the cells that were expanded at ISS. So no impact really on proliferation as far as we can tell. We also look at, of course, we are talking about cell expansion, so we look at cell cycles and look for expression of cell cycle checkpoints, inhibitors. So CDKN2 is a cyclin-dependent kinase 2A, which inhibit transition from G1 to S phase. And then we also look at E2F1, which is interact with RB retinoblastoma protein, which is a powerful cell cycle regulator. And then the polo-like kinase 1, which is serine threonine kinase, which inhibit transition from G2 to M phase of cell cycle. As you can see, at 1 week culture, there is really no significant difference. So also in 2 weeks culture, other than the PLK1 is significantly decreased compared to the ground control. We really think more studies needs to be done, but clearly it looks like microgravity might be inhibiting the MSC expansion or progression in the cell cycle. But I think more studies need to be done. And then we look at differentiation, both using using stain for osteogenic differentiation and the oil red for adiposogenic differentiation. The figure A on the top is just showing normal cell grown in regular media and then the differentiation media. We also look at genes that or transcription factors that are known to be associated with osteogenic differentiation, which is the first two, the CLK1A and the RANKS X2, and then the CRAD B is for adipogenic differentiation. And the summary of it is that we haven't seen any difference between control and space-grown MSC in their ability to differentiate. We also look at conditioned media for cytokine release, looking at both ISS grown and ground control. And the interesting thing is that we look at 65 different cytokines, and we were only able to detect significant difference in two. There's CD40 ligand, so which is significantly decreased, and then the platelet-derived growth factor is significantly increased after 1 week of culture. But we see more cytokines being affected after 2 weeks of culture. For example, the IL-10 is significantly decreased. The MCP3 also significantly decreased. So as the SDF, which is a powerful chemokine for attracting cells and impacting on cell migration, that also is decreased. This was previously reported, so it's interesting to see our studies also confirm previous report. And then also the IP10 is increased. I think, again, with this one, the conclusion you can only draw on this is that, yeah, cytokine and growth factor expression is impacted by the absence of gravity. But more studies would be needed to really comprehend the downstream effect of these changes. We also look at potency, assay. We wanted to see if the MSC are more immunosuppressant than when they were grown at under microgravity condition. MSCs are known to be immunomodulatory and actually it's one of the primary reasons that they have been used. So they are used in treating rejection of organs, also used in as immunosuppressant to control graft versus host disease after bone marrow transplant. So we wanted to see whether that properties of theirs is maintained or enhanced or decreased. And the short answer is the MSCs that are expanded in space are much more immunosuppressive than the control, both at 1 week and 2 weeks. Finally, we come to safety. We are worried about DNA damage. So we use this marker that is this transcription factor is part of the histone family of transcription factors that get exposed if the DNA is damaged. So we stain the cells, and then we also expose the cells with etoposide, which has DNA damaging properties as our positive control. We also use DAPI to stain the nucleus so that we wanted to show that the damage, or the DNA damage, is in the nucleus. So basically, you can see when the drug is used, you can see a lot of DNA damage, and without the drug, there is no damage. And we haven't seen difference between gram control and space for all the weeks of culture. We also did chromosomal studies and karyotyping. This is a gene banding, and clearly we didn't see any chromosomal abnormalities when we compare the ground control and the ISS. And then finally, we said, well, what about tumor formation? So here this is a positive control. We're using a fibroblast sarcoma cell line. You can see the colony being formed, and this is what we are looking for. So, and then this is a negative control of just the cells, different from the ones that are used in the control group. So you can see the ground in 1 week, and then the microgravity ISS in 1 week, and then in 2 weeks. Both, there is no evidence of tumor formation like what we have seen in the positive control. So the summary of our finding is that, so we asked fundamental question: do MSC survive and grow faster in space under microgravity condition? And the short answer, yes, MSC grow in space under microgravity condition, and their growth is comparable to the one on the ground control. So we didn't see that further expansion where we can grow more cells, but these are the MSCs. We've seen some expansion on the HSC, and there wouldn't be time for me to go into that data, more to come down the road. And then in terms of feasibility, growing MSC in a sterile and safe way at the International Space Station, clearly we can see that we can do that. MSC can be cultured without any microbial contamination. We show that they haven't been transformed to form tumors. They have normal chromosome. The karyotype is normal. No DNA damage seen. We also have seen that there is an effect of the microgravity on the MSC capacity to secrete some cytokine and growth factors, and that the MSC actually that they are more immunosuppressive compared to the ground control. So those are the main findings. Obviously, we have a lot more questions, so it's really nice to be given this opportunity, and thank you so much. I will stop now. Thank you.
Thank you, Dr. Zubair. This was fascinating. Your results are very intriguing, and looking forward to seeing more of them. I want to remind everybody that as you come up with questions, please enter them into the questions tab at the end of during which we will have a panel session. We will start addressing the questions. Our next speaker is Jana Studemeyer. As the Commercial Innovation Officer at Space Tango, Jana Studemeyer leads the commercial market creation for biomedical and technology manufacturing applications in low Earth orbit. Today, you'll hear about Space Tango's work to expand capabilities and partnerships for stem cell discovery and manufacturing applications in microgravity. Go ahead, Jana.
Good morning, Liz, and good morning, everyone. Thank you so much for this opportunity to present to the group today. And thank you, Dr. Zubair, for an excellent opening presentation. I'm looking forward to hearing more from Dr. Sharma and from Dr. Shepard and Dr. Lin later on from CERM as well. I want to start by just saying, you know, it's really an exciting time, both in space and on the ground for regenerative medicine. And so I'm going to walk you through this morning a little bit of capabilities development that we're doing to support more sophisticated biology that really will help us to understand that regenerative medicine opportunity in microgravity, as well as some of the changes that are taking place on the space side that are helping us as well. So at Space Tango, our focus, as you probably know, I think most of you are aware of the work that Space Tango is doing, but we're really looking to inspire, innovate, and create a better future for humanity, utilizing the unique environment of space. And our mission is really to manufacture health and technology products in space that create value and transformational solutions for humanity. So we're very focused on opportunities to not only understand the fundamental science, but to look at how we can build a robust commercial space economy that has a new market segment in it for biomedical products. In terms of the automated systems, so we put together facilities that really support iterative R&D and manufacturing on the International Space Station. And part of the focus for us is to be looking at sort of, you know, more than one opportunity to be able to understand some of the complicated biology. It's complicated on the ground, and it's even more complicated to sort out in space, as you were just hearing from Dr. Zubair. Just briefly, I think everyone probably knows we're located, our headquarters are in Lexington, Kentucky. I'm actually based in San Diego, California. We have also part of our team in Washington, D.C. for strategic engagement with our congressional representatives, and we have ground operations in our Houston facility, satellite facility in Houston, Texas. Launch operations are performed out of NASA Kennedy Space Center at Cape Canaveral and Wallops Flight Facility, Wallops Island, Virginia. And on ISS, we actually have our 2 Tango Lab facilities and our power descent utility locker, which I'm going to talk a little bit more about in this presentation, but it's a newer capability that has been developed by Space Tango to allow us to have power on ascent so that we can actually maintain the biology as we transit to the International Space Station, as well as while we're on orbit. So you heard a little bit about rockets, and I know that we talked, that Dr. Zubair mentioned the splashdown. Just for background here, for anyone who is not aware, there are 2 providers for commercial resupply missions, which is where the science that goes up actually flies in. One is SpaceX, the Falcon 9 and the Dragon capsule, which everyone I think has seen quite recently, even with American astronauts on American rockets from American soil, which is fantastic. And Northrop Grumman also has the Cygnus capsule, the Antares rocket and the Cygnus capsule. Just a little bit of background here for anyone who is not familiar with these vehicles and space. The Dragon capsule is the only one that returns back to Earth with samples in it. The Cygnus vehicle goes to the ISS, delivers science and supplies, but actually is the trash vehicle that is designed to burn up on re-entry. So when we talk about biological payloads, and you know, Dr. Zubair mentioned, SpaceX is really the only capsule where those samples can return. So often we try to fly biological payloads on a SpaceX mission because they can go up and back in the same vehicle. You can actually go up on a Cygnus vehicle and come back on a Dragon. but there's a little bit of time between those launches. And so the sensitivity of these payloads, we try as much as we can to put them in the same mission. The exciting part of what's happening on the space side is that in addition to those two vehicles, we are now having new vehicles come online and new opportunities for flights. So where it's been difficult and sometimes a long time frame to be able to get to an opportunity for launch, these additional vehicles and providers will be able to give us an opportunity to really have the increased access that we need. There's also some future commercial facilities that are being developed. So ISS, as everyone knows, this is the 20th anniversary year. It's been in orbit for quite a while as a laboratory, but recently NASA actually awarded the docking port to Axiom Space for a commercial space station that will be used to really maintain our presence in low Earth orbit once the ISS is decommissioned. Space Tango is actually working also on our own Free Flyer, an automated facility that we will be able to use for manufacturing on orbit of both technology and biomedical products. But it will give us the ability to really be on a schedule that will be orbiting, we won't be docking, there won't be people on there, so we can kind of control the timeframes of up and back in microgravity. So that's a little bit about capabilities, and it's good to see the increased access. But I want to switch gears a little bit and talk now about what it looks like to do research in space and what kinds of capabilities exist. So I thought I would start first with just sort of the basic overview for an understanding of what a lab actually looks like in space. On the top here, you see your typical lab bench with everything out on it and lots of bottles and reagents and all kinds of equipment. On the bottom, you see the inside of the ISS. Everything is bolted to a wall or put into a locker format, obviously so that it doesn't float away. And this really is one of the big differences about working in space versus working on the ground. Simple things that you would do in the laboratory, like a media exchange, that would take maybe 45 minutes, in space manually takes several hours. So it's really a very different process. Our facilities on ISS, we have two Tango Lab facilities, as I mentioned. The first one was actually installed in 2016, and the second facility was actually put up in 2017. So these facilities give us the ability to collect data from payloads that are automated and remotely control those payloads from the ground. It removes the astronaut hands and time that is very expensive and can at times be difficult to manage. The crew's schedule, as you may be aware, is very busy while they're on ISS, not only the research that they're doing, but the work that they're doing on station and the exercise that they need to do just to maintain their own physiology. So when we think about ways that we can help to accelerate that science, automating it and taking astronaut hands out of the process is something that we see can really help. The Power Descent Utility Locker, which is shown in the bottom picture here, is the locker that I mentioned at the beginning that we created to help us on ascent really control the conditions that you need to maintain cell culture on the way to the ISS. And once this facility is installed on board, we can downlink the data that we retrieved during the ascent of the vehicle and actually continue the experiment. Just a brief mention here of the facilities and sort of the flexibility. You know, one of the things that we also did with our Tango Lab facility was to take it out of its locker position and put it into a docked capsule, the Cygnus capsule, while it was docked to the International Space Station, really showing that we can kind of extend the laboratory that exists. Space is at a premium on ISS, as you can tell from the pictures. When you see everything, you know, every space is used. So to have the opportunity to be able to extend the laboratory facilities to a vehicle that's docked to station also increases our capability. This is just a short video of what the actual uninstall. These are two of our CubeLab automated facilities, which I'll give you a little more detail on in just a minute, that are being removed from that Paul facility and put into cold storage. So each of these CubeLabs contain, this is one of our tissue check payloads for the NIH, and each of these CubeLabs contain science that has been now conducted, long duration cell culture on station, and now being moved into cold storage for return. So a little bit about what's inside the CubeLabs. And as I mentioned before, the automation for us is something that we understand to be able to support the sophisticated biology that is now a focus of regenerative medicine. We really need to have systems that allow us to look at different configurations of cells that can be grown in an automated way. So the CubeLabs that we've designed they permit long duration cell culture for things like tissue chips. We have 6, 12, and 96 well plate configurations, as well as bag configurations. And we've added to the CubeLab an automated microscopy system. This is a great group from San Diego, and I have to sort of give a shout out to San Diego because they are local, but Etaluma is a company that MSC actually have optics from Etaluma. It's a perfect scope for ISS because of the fact that it is not only very compact and can fit into the CubeLab, but it also has a unique vibration resistance. And so we're really excited about increasing capabilities to do on-orbit measurements with a scope like this in an automated way. So let's talk a little bit about translating ground science to space science. And when I talk to the teams that we work with, I always talk about the fact that it's not really just the translation of ground science to space science. In our case, it's the translation of ground science to automation and then to space science. And there's lots of things that we need to think about. So the conditions on ascent, for example. What's the metabolic state of your cells, and how do you want them to arrive at the ISS so that you can really make sure that the data that you're capturing is data from a microgravity environment. The conditions on orbit, the timing for evaluating the desired effects of microgravity that you're looking for. So media samples and reagent storage, temperatures that need to be maintained for conditioned media that has growth factors in them within the same automated system as an incubator that keeps the temperature of the cells at 37 degrees. And then, of course, the sampling and imaging time points and how we can download that data and look at that during the course of the cell culture that goes on orbit so that we're able to make any adjustments that might be needed in near real time. And then the return of your cells. So fixed, preserved, frozen, live, those are all capabilities that we have. And in conjunction with doing on-orbit measurements, we want to make sure that we bring back cells that allow some additional post-flight analysis as well. In terms of the cell culture capabilities that the automated hardware we're developing is designed to provide, so NASA safety certified, obviously the systems that go up on ISS are at least 2 levels of containment. They're all built for biocompatibility and designed for sterilization of the components before the flight build. They have the ability to control temperatures, standard conditions for cell culture, 37 degrees, 5% CO2, do automated media exchange, media sampling over a time course, imaging, drug challenge, if that's something that you want to look at as well. And then obviously the fixation and cryopreservation or live return of cells. We've also, because of COVID-19, developed some additional benchtop capabilities. Everybody knows it's a little bit difficult to be in the lab right now. And those that are in the lab, for us to be able to visit and sort of be right there with some of the scientists, that's been a little challenging under the current travel restrictions as well. So what we've done is put together a benchtop capability that really allows the basic sort of cell feeding and agitation that you would see within the CubeLab while it's on orbit, to be remotely controlled from the ground. So we have a web-based GUI for our partners to control and for us to control from Space Tango at our remote access points. And we have data that we can pull from that and put into a portal that we're able to share with the researchers, and we can all watch as we're working through the validation of these systems to make sure that they're working the way they need to. So the focus on automation for us, there's a lot of reasons why. In addition to the fact that astronaut time is expensive and sometimes difficult, to make a change on a near real time is often a little tricky and challenging because the crew schedules are so full, but also for the consistency. We're very lucky to have people like Peggy Whitson, and you're going to hear from Dr. Sharma next, I think, about the work that was done by Kate Rubens, which is phenomenal. And someone like Kate Rubens is an amazing biologist here on the ground, but not every astronaut that is on station is Peggy Whitson or Kate Rubens. And so we feel like the automated systems not only give us the ability to have higher throughput, but also the improved consistency. And we really focus a lot on making sure that those cube labs really have temperature, pressure, gas concentration, pumping speed, imaging, and other types of sensors that standardize the science environment. We take a step-by-step approach to product development. The automation allows for on-orbit tests of sub-technologies, so we really will look at each piece of the different part of the payload that's going to be automated, and sometimes we actually even look at that in a microgravity environment with things like beads or other ways to simulate before we actually put cells in and fly it. There's never enough testing that can be done on the ground to get ready for these types of experiments. The translation, as I mentioned, from bench to automation really does require that we grow the cells in the system and make sure that not only the engineering design meets the requirements, but that the biological requirements are met as well. And the ability for the near real-time data monitoring that I mentioned, downlinking data to our remote station allows us to have the ability to even do things like make changes to the ground controls. So we have options for doing either parallel controls with a slight offset so that we can pull that data from what's happening on orbit and make adjustments if we need to, or at times some payloads actually an asynchronous control, done after the work is done on station to match those conditions exactly, makes sense. Most of the biological payloads, obviously you want to load them with the same cells that you're flying so that you limit the variability there, but there are opportunities to look at controls kind of from both perspectives. So now you know a little bit about what the hardware looks like. What happens when we actually now have tested the system and are ready to fly? We have pre-flight loading that goes on at Kennedy Space Center. So most of our teams will come down and be with us for several days before the launch, getting ready. And as anyone who has flown knows, launches are always called launch attempts because they can sometimes move. And with biological payloads, you really need to be prepared to have other sets of cells that are available to you in case there's a launch slip. Once the actual pre-flight loading is completed, the payload is then turned over to NASA. It is loaded into the rocket and actually sent to the International Space Station. Once the vehicle docks, it's unloaded, and the cube labs are then attached to payload cards that give us that remote capability to control and see what's happening on the ground while we're on orbit, and loaded into the Tango Lab facilities. Once the ground control operations and the cell culture period is completed, then the reverse happens. The cube labs, as you saw in that video, are actually detached and returned to us in the vehicle, and then splashdown, payload return. As Dr. Zubair mentioned, we were picking payloads up since I'm here in California, right at Long Beach, and now this year we're going to actually be in the Atlantic, so closer to the Kennedy Space Center, where there are facilities to actually take your payloads and samples and right at return into those same facilities where you loaded previously. Just briefly, so we've 17 missions so far since our Tango Labs were installed in 2016 and 2017, 88 payloads and 156 experiments. And I'm just going to briefly give you an overview of the types of projects that we're working on right now. So I mentioned NIH tissue chips in space. We have a partnership with Emulate doing a blood-brain barrier chip. We also, in the tissue chips in space 2.0, are working with Emulate on a gut chip. And we also have two other projects, one with University of Pennsylvania and Children's Hospital of Philadelphia for bone marrow and lung, and one with University of Florida, Gainesville for skeletal muscle. Really excited. These payloads are coming up on SpaceX 21. So we're going to be very excited to see a lot of this work starting to bring data back to us as well. In terms of the NSF tissue engineering collaboration that CASES has put together with NSF, we have 3 payloads that we are actually supporting. One from Mount Sinai on cardiac stiffness, one from University of Michigan on mechanotransduction of osteoblasts, and then one from University of Texas and Texas Tech that is actually looking at cardiac organoids. So we're very excited to be working on several of the projects with NIH and NSF. And recently, we also flew brain organoid models, which you may have seen some information on these payloads as well. We're very excited to work with National Stem Cell Foundation and also with University of California and Arthur C. Clarke Center for Human Imagination. These 2 organoid payloads, one actually looked at neurodevelopmental biology, so very early stage organoids, and the other one looked at neurodegenerative disease using iPS cells from patients with Parkinson's and primary progressive MS. So in addition to the more 2-dimensional tissue chip models, moving up to 3-dimensional stem cell models is something that, you know, again, takes the biology to yet another level of sophistication. And recently, in April of this year, we actually were very pleased to be awarded by NASA 3 projects that are really focused on commercialization for regenerative medicine. You know, the support of the public-private partnerships is really what will drive building that market 250 miles up and expanding that definition of global. And these are really exciting projects. I'm putting all 3 of them here, but the 2 that are stem cell based, you've probably heard about our artificial retina project, which is a protein polymer, but also we're looking at stem cell translational medicine with UCSD Sanford Consortium for Regenerative Medicine, and then Cedars-Sinai looking at parameter setting studies for stem cell manufacturing. So these are all really exciting and part of the foundation that we're laying for these types of products with the, you know, opportunities to manufacture is setting up GMP capabilities for manufacturing as well. So to close, you know, we really are on the brink of returning to space, and this time it's going to be to stay. So I'm super excited to have the opportunity to work with all of the teams that we work with, the great science that's being done, for Space Tango to continue to contribute capabilities that are automated that support sophisticated science.
Thank you, Jana. As you can tell, Space Tango has thought a lot about how to conduct research in low Earth orbit. Again, if you enter your questions in the questions tab, we'll get to them during the panel session. Our next speaker is Arun Sharma, stem cell and heart biologist at Cedars-Sinai Medical Center's Regenerative Medicine Institute. Arun will tell us about using human pluripotent stem cells to model the effects of spaceflight on the heart, a paper that was published late last year and was conducted on orbit just about 4 years ago this month. Arun.
Well, thank you for that kind introduction, Liz, and thanks to the National Lab for giving me the opportunity to talk a little bit about our work that we've done over the last few years utilizing human pluripotent stem cells to model the effects of spaceflight on the heart. And I'll be recapping some of the work that I conducted as a graduate student at Stanford University in the Cardiovascular Institute, but I'll also briefly touch on some of the ongoing work that we're doing here at Cedars-Sinai. So the cardiovascular effects of spaceflight, why are we actually interested in doing this work? Well, we have an understanding as to what happens to the heart as a whole, as a tissue, during extended microgravity. There's an understanding that there's a reduction in overall cardiac work, as well as an increase in heart muscle loss. And this is as a result of that reduced cardiac work. And also as a result from that muscle loss, there's a shape change that actually occurs in the heart. The heart actually assumes more of a spherical shape on orbit as opposed to the traditionally fist-shaped structure of the heart. So we have an understanding of what happens to the heart at the organ level, but we have less of an understanding of what actually happens to the heart at the cellular level. And so we are using a couple of revolutionary pieces of technology in stem cell biology to really help address some of these issues and help us better understand what happens to the body at the cellular level in microgravity. Okay, so we are using induced pluripotent stem cells, and for those of you who are on this call and have an interest in stem cell biology, these cells are of course no surprise and nothing new to you, but just as a quick recap, induced pluripotent stem cells are a cell type that is largely equivalent to the embryonic stem cell. These are cells that are able to be produced from a small sample of an individual's own skin or blood. They can be reprogrammed to induce pluripotent stem cell colonies, and then from there differentiated into a variety of different cell types, such as cardiomyocytes, the muscular cells of the heart. And of course, the fun thing to note about these cells is that since they're cardiomyocytes contracting heart muscle cells, they're able to spontaneously contract in a dish. There are a number of different applications for these cells, including potentially for autologous cell therapy. But the focus of this presentation and the focus of most of my work has been on using these cells for disease modeling, as well as for drug screening and drug discovery, and using these cells for examining the effects of microgravity as an analog for the human heart. So I'll give a brief overview of some of the work that we did over the last few years. We had the good fortune to receive some funding from the Center for the Advancement of Science and Space and CASIS to derive iPSCs, induced pluripotent stem cells, from 3 separate individuals, and then differentiate those cells into contracting cardiomyocytes, then transport those cells to the International Space Station via a SpaceX Falcon 9 rocket. Of course, we also had a parallel set of ground-side control samples here on Earth so that we would be able to compare the effect of microgravity on our iPS cardiomyocytes on space to a parallel set of conditions. Once we were on orbit, we conducted a number of analyses, including gene expression analyses, looking at how these cells change in terms of their gene expression, as well as functional analyses for contractility, calcium imaging, and structural cellular analyses using immunofluorescence. We also returned a sample of these cells back live to the planet for further analyses, and these results were actually recently published in stem cell reports at the end of last year, so feel free to take a look. I'll give a brief overview of our approach to actually culturing these cells, and also I'll talk a little bit about some of the data that we got from our experiment. This was a very collaborative effort, and our implementation partner in Bioserve Space Technologies helped us develop a custom set of cell culture growth plates. These are fully enclosed 6-well plates that have individual wells isolated to prevent cross-contamination. They're fully enclosed, of course, because we want to avoid media and nutrients and cells from escaping aboard the microgravity environment of the International Space Station. So these were the culture systems that we actually used aboard the International Space Station. Perhaps the greatest assistance and the greatest help that we had for this entire project was from Dr. Kate Rubins, who is not only a NASA astronaut, but also a very talented cell biologist, as Jana alluded to in her talk. We had the very good fortune of working with Dr. Rubins over the course of a month in the summer of 2016, so that we could actually examine the effect of microgravity on these iPS cardiomyocytes. And what Dr. Rubins was able to do was to conduct weekly nutrient exchanges on these cells to maintain their viability, also conduct microscopy, and maintain the iPS cardiomyocytes for 30 days aboard the space station. So what do we find? I'll walk through some of the data and some of the results that we actually obtained from these experiments. Well, first we found at the macro level, the iPS cardiomyocytes did not exhibit significant overt changes in cell morphology or sarcomere structure after return from spaceflight. Sarcomeres are the individual contractile components of cardiomyocytes that actually enable them to contract at the cellular level. As you can see here in this video, the iPS cardiomyocytes did continue to contract during prolonged exposure to microgravity, although there were some changes in the way the cells actually contracted on orbit. So this was the first demonstration of long-term cell culture aboard the International Space Station. Again, as I mentioned, we cultured these cells for 30 days. And one other thing to note is that these are relatively advanced cells. As you can see here, they're functionally active, they're contracting, they're electrophysiologically active cells. So another important aspect of this project was to demonstrate that long-term cell culture is indeed feasible aboard the International Space Station. As I mentioned, we observed some changes in the way the cells actually contracted on orbit. There was a trend downward in terms of their overall beat rate after up to 2.5 weeks on flight. There's also a trend downward in their contraction and relaxation velocities as well. And so this may be a result of the cells adapting somewhat to their microgravity environment. These are functional cells, as you can obviously see through the contraction of the cells. And to elicit the contraction, cardiomyocytes have to undergo an excitation contraction coupling process, whereby the calcium ions within the cells are actually activated, and ultimately these ions are what lead to the individual contractions of the cardiomyocyte. And because of that reason, you can track calcium flow and calcium flux in individual cardiomyocytes. So we did calcium imaging on these space flow and heart cells after they returned back to the planet as another functional indication of how the cells are behaving. And what we did observe is that these cells exhibit reduced calcium cycling after return from spaceflight. Finally, we're able to preserve a sample of iPS cardiomyocytes at multiple time points during spaceflight for RNA sequencing. So this is conducting gene expression analysis of space-flown samples and compare them to ground-side controls as well as post-flight samples, samples that were preserved after the cells returned back to the planet. And what you can see is that the samples that were returned to the planet in the post-flight set of samples here largely show a similar gene expression profile to the ground-side controls, whereas the flight samples exhibit a slightly different gene expression profile based on the cells that are activated, based on the genes that are activated in these cells. And if we took a closer look at some of the gene pathways that are actually altered in those space-flown samples, we identified a number of genes related to mitochondrial and metabolic function. So to conclude, we were able to demonstrate that human iPS cardiomyocytes, human heart cells, could be successfully cultured long-term aboard the International Space Station. As I mentioned, this was the first demonstration of long-term cell culture aboard the International Space Station, and also culture of a complex cell type, a functionally active, electrophysiologically active cell type in human cardiomyocytes. These cells exhibited some alterations in contractility within weeks of spaceflight, although it did not change their overall structure as shown by immunofluorescence. They also showed gene expression changes, and in particular, changes in pathways related to mitochondrial and metabolic function. And ultimately, we believe that this is a useful model system that could be used to model the effects of spaceflight and microgravity on the human heart at the cellular level. So I'll leave you with some future directions, projects that are in the works, and some food for thought for additional analyses that will need to be done. How can we improve this particular experiment and this particular model? Well, this was a two-dimensional cell culture system. Everything that I showed you, all the data that I showed you, was from two-dimensional cardiomyocytes grown in a monolayer. Ultimately, the human heart has a variety of different cell types that are important to its function, not only the contracting heart muscle cells, but also endothelial cells, the support fibroblast cells, and so on. And a better model system, a better multi-lineage model system would be able to leverage these other cell types as well. There are a variety of different improved model systems that are being developed right now utilizing, for example, tissue engineering approaches that can artificially print multiple cell types into a single system. Organ chip approaches that Jana talked about briefly, where you can grow multiple cell types on an integrated organ chip which is connected through a pseudovasculature, and also organoids, these three-dimensional collections of stem cell derived tissues, would also be a useful model system to better understand the effect of microgravity on the human heart. Additionally, there's a need for an automation of processes. Again, as Jana alluded to in her talk, crew time is very precious on the International Space Station. And although we were very fortunate to have an astronaut working with us, and Dr. Kate Rubens, who has a background in cell biology, that's not always the case. Of course, the astronauts have a variety of other tasks to attend to while they're on the International Space Station. They have a variety of different experiments that they're doing for other projects, and also maintenance of the station itself. So if we can further facilitate the automation of some of these cell culture processes for maintaining these cells on orbit, that would be advantageous not only for the astronauts, but also for the scientists as well. And finally, scalability and in-orbit production. Is this something that would be feasible? There is some interest in potentially utilizing the station in microgravity environments as a means to facilitate the production of stem cell populations and stem cell derivatives. This is something that you would have to examine in the context of throughput as well. Can we manufacture these cells at a GMP or good manufacturing practices level, so that we could biomanufacture these cells and potentially use them for translational purposes back on the planet? And we've actually had the very good fortune to obtain a recent grant from NASA to further investigate this. This is with our team here at Cedars-Sinai in collaboration with Space Tango, who has been developing a number of these automated platforms for facilitating cell culture on station. And so we're in the process of commencing that particular project. And with that, I'd like to thank everyone who has been instrumental in helping to get these projects off the ground, no pun intended. Of course, collaborators at the Stanford Cardiovascular Institute for the two-dimensional iPS cardiomyocyte projects, as well as collaborators here at Cedars-Sinai and Space Tango, our implementation partners in BioServe, SpaceX, and of course the Center for the Advancement of Science in Space, which has helped fund this particular work. And for additional information on our iPS cardiomyocyte project, our two-dimensional iPS cardiomyocyte project, I would refer you to our recent publication in Stem Cell Reports. So thank you again for your time.
Fantastic. Thank you, Arun. You know, that first heart cells experiment you described was really a pathfinder for the field in space, and now we see that you're very much involved in the future as well. Next, we have Kelly Shepard and Stephen Lin from the California Institute for Regenerative Medicine, CIRM. Kelly is the Associate Director of Discovery and Translation, and Stephen is a Senior Science Officer. The mission of CIRM is to accelerate stem cell treatments to patients with unmet medical needs. And today, we will hear more about CIRM and the use of stem cells for predictive modeling and therapy.
Thank you very much. Everybody can hear me, I assume. I'm going to be asking for help with advancing my slides because my connection may be a little bit slow. So I'll try to remember to ask for that. Anyway, I want to begin by thanking you for inviting us. Stephen and I are program officers, as Liz mentioned, at the California Institute for Regenerative Medicine, or CIRM, which is a California state agency that was established by a ballot initiative in 2004 to fund stem cell research in California. Primarily, it was established to support the use of human embryonic stem cells, but it actually supports stem cell research in a much wider variety of areas. And we've been funding over 1,000 projects since 2004, over the past 15 years. Is tag team our presentation a little bit today. I'm just going to start things off by providing a bit of background on our activities over the past 5 years or so, since this is more relevant to what we're talking about today. And this will provide some context for how our perspective has been informed by what we see as current issues and needs in the stem cell field. And then after I go over that, I'm going to transition to Stephen, who will provide some specific anecdotes and examples of some projects from CIRM investigators and the relationship to projects that have been done on the space station thus far and how they might be further informed by such studies. Okay, next slide, please. So as Liz introduced, the mission of CIRM is to accelerate treatments to patients with unmet medical needs. And our goal is to use stem cells in any way possible to do this. Next slide, please. Specifically, that means we fund research and development. And we focus our research in 5 core areas, which are depicted on this bridge. Sorry, I forgot to say change slides, didn't I? I won't really be talking much about our educational pillar today, but I will be telling you primarily about some of the work done through our first core pillars here, infrastructure, which is to create resources for the scientific community to use. And then our pipeline programs, which fall under 3 different stages of maturity: discovery, translational, and clinical. So by discovery, I mean the earliest stage of research. And we have supported both from the very get-go, no preliminary data, hypothesis testing, new ideas, figuring out what happens, how to make things work, to candidate discovery, looking for something that can be used as a potential therapy or as a potential tool for identifying therapies, and knowledge generation. The translational stage is where somebody's made a promising finding in the lab, a development candidate, and they need to take it through the steps that is necessary to begin discussions with the FDA about bringing to a clinical trial. And then, of course, there's the clinical stage, which to CIRM begins at IND enabling, which is after you have agreement with the FDA on a promising set of studies that could lead to an IND filing, to phase 1, 2, and 3 clinical trials. And ultimately, we don't have anything at the stage yet, but ultimately, hopefully, a drug approval or commercialization. Next slide, please. So the types of projects that fall into these pipelines are quite diverse, as you can see. They cover a wide range of activities, but they kind of fall down into 3 basic areas. So knowledge generation, using stem cells to understand basic human development and embryology, to understand the basis of human disease, to discover new disease targets. We also support a variety of projects where stem cells are used as a tool of discovery. So for example, to develop a screening platform for drugs, to model disease in a dish, these types of studies. But we don't just fund stem cells as a tool, we also fund traditional tool development that can be used to enable the stem cell field, such as novel imaging modalities, new cell editing technologies, CRISPR-Cas9, these sorts of things, directed towards editing stem cells. So basically funding tools and technologies that can make stem cells more useful. We're also interested in developing things that can make automated processes, because ultimately to bring therapies based on stem cells to the clinic, we need to make them more expensive and practical, reproducible, and consistent. And so I see a lot of common ground there in the needs for what it takes to bring a study into space to what it takes to bring a study into the clinic. And so next slide, please. When we think about the types of these projects that I've just shown on the slide built across this pipeline going from the earliest stage to the clinical stage, you're going to see a lot of common ground, as I said, with some of the types of studies that are being conducted in space and being contemplated, and some of the studies indeed that we've heard about today. So at the very early end of the spectrum, some of the most significant questions then when CERN was founded and now relate to the rationale. If you want to develop an application for a stem cell, what is the right stem cell type to use, or derivative of a stem cell? Do we know what our target cell is? Do we know enough about human biology to know which the right cell is to study or is responsible for a disease? Is it one cell or is it a group of cells together that are necessary, right? So even if you know that cell, how do you procure that cell? A lot of the work that CERN funds are trying to procure these cells by differentiating them from a pluripotent cell starting point. That's a scientific process of discovery to be worked out on its own. In addition, stem cells can be obtained from tissues, from adult tissues, but being able to study human stem cells, such as those, which have to be removed and propagated in the lab, there are a whole bunch of challenges around culturing and maintaining them in the lab while they maintain their potential and functionality. It's very hard for many adult stem cells to maintain their ability to be expanded or even activated outside of their niche, which is normally in a tissue inside of an adult human organism. So other considerations that we have to think about, of course, even at the earliest stage, are donor source and eligibility. Eligibility, because if you're going to eventually use one of these cells as a therapy, it has to be consented appropriately to get through the FDA approval process and to be ethically sound. Donor source, we have genetic background of the donor that comes into play. This is also important if you're interested in studying disease processes. You want to, as Arun mentioned in his talk, the possibility of taking cells from individuals with specific diseases so that you can model them in vitro. So those types of considerations. As we move along to more mature science, getting into translation or early development and eventually clinical trials, the challenges become quite different. So at this point, you've already identified a promising stem cell-based candidate that you want to eventually test in a more broad use application. Here are the challenges developing, and we heard about this a little bit in our first presentation as well, is the scaling up. You're going to have to go from a dose that might have worked in a rodent to a dose that is going to be effective in a person. So you're going to have to be able to grow the cells in much larger numbers while maintaining their functionality and potential. And not only that, you're going to have to change your process so that it meets GMP compatibility requirements, so that it can be produced in a gene compatible manner. And that can change, that can have effects on your cell. So a great deal of challenge, especially in our field, is centered in this space right here and right now, and that's developing the GMP compatible process and the scaling up process. And although some projects have made it into clinical trials, of course, there are a whole new set of challenges that they're facing and navigating now and will continue to do so. And there's even more scale up that will need to happen if this is going to be something that is going to be offered as an approved drug to a more widespread disease. You can't just make enough cells for one patient. You are going to want to treat a lot of patients. So you have to be able to develop a commercial scalable process. You have to figure out how are you going to store and ship your cells. And all of this, you have to maintain the integrity of the cells. And so we're not quite there for most of the types of projects that CERM funds, but these are the challenges that we see as looming on the horizon. Okay, next slide, please. So this is the state of the field today. There are a number of stem cell projects that are in clinical stage trials or very close. The more mature therapies that have been worked out longer are the mesenchymal stem cell products. And we also have a long history of bone marrow transplants now, which are basically hematopoietic stem cell transplants. There are also some examples of some primary neural stem cells in the clinical trial, as well as immune progenitors, such as T cells and dendritic cell progenitors, and other tissue-specific progenitor cells. There are also now some pluripotent stem cell-based products that are also in clinical trials. You may be aware of some of these. We have pancreatic progenitors. There are some cardiomyocytes derived from human embryonic stem cells, neural progenitors, dendritic cells, retinal derivatives. The ones with little asterisks are the ones that are currently in clinical trials. The others are getting very close. But as you can see, the field is evolving, and a lot of the technology that was in its infancy 15 years ago is getting ready to a stage where these questions and challenges are a reality, and they need to be addressed now. Next slide, please. So this is just a snapshot of some of the earlier stage projects that CERM's been funding over the past 5 years or so through our candidate discovery program and our hypothesis testing program, which we call DIS1 and DIS2. So you can see on the left that about half of our projects are dedicated towards trying to find a therapeutic candidate, either from stem cells or that can be discovered through the use of stem cells. We still fund a fair amount of projects doing basic knowledge building, hypothesis testing, and about a third of our projects are devoted towards developing stem cell-based tools or technologies, or, as I said, imaging technologies that can be enabling for the stem cell field. Although the majority of our early stage programs are still focused on pluripotent stem cells, primarily embryonic stem cells, induced pluripotent stem cells, or using both, we do have a fair amount of projects looking at hematopoietic stem cells and a whole variety of other adult stem cell types. And we also have some projects who are looking at direct reprogramming, which is skipping the pluripotent stem cell step and trying to convert one differentiated stem cell type into a different fate, to sort of avoid some of the challenges that come with working with pluripotent stem cells. Next slide, please. As we move down that pipeline, as you can see, our translation stage, we still fund a fair amount of pluripotent stem cells there. Again, these are projects that are engaged in studies that will allow them to bring a package to the FDA for discussion. Now, you can see that a good portion of these are hematopoietic stem cell projects, but these aren't just your ordinary bone marrow transplant. These are gene-modified hematopoietic stem cells. So CRISPR-Cas9 edited or lentiviral modified. So these are kind of the next frontier of stem cell biology. So there are also a lot of challenges to be worked about to get efficient editing of these stem cells so that they can be used in a safe and practical manner. Next slide, please. And this is the last slide I'm going to share before I transition to Stephen, because what I want you to see is that there really is a lot of momentum in our field building for the pluripotent stem cell derived therapeutic products, as well as what I just alluded to, gene-modified versions of established stem cell products, such as hematopoietic stem cells. But as you can see from this kind of green coloring and on the right here, even these darker green colors here, even a lot of the pluripotent stem cell products that we're funding are gene-modified. And as you can see, these are primarily the allogeneic candidates, meaning donor-derived. And as you can imagine, or you might guess, a lot of that gene modification is an attempt to make a universal cell product, something that can be used by people with different immune backgrounds. Now, you've talked a little bit about manufacturing cell therapies in space, and I think that probably makes the most sense for a product where you could produce a bunch of it and bring it back and have it be an off-the-shelf therapeutic. So figuring out how to create immune compatible cell types that could be manufactured safely and effectively and inexpensively is going to be something that's extremely valuable for the field as a whole. So I think from here, I'm going to transition to Stephen now, and he can give you a few more concrete examples of some of what I've just shared with you in our overview.
Great, thank you, Kelly. Can you advance to the next slide? Okay, so as Kelly described, CIRM has established a pipeline of awards that span from basic research and discovery to clinical stage programs. And so I'm going to describe several examples of CIRM investigators that are developing therapeutic products at different stages in CIRM's pipeline, but they've also conducted projects aboard the ISS or are planning to do so. And actually there's some overlap from the previous presentations to some of the examples I'll give. And so I'll also describe some scientific questions related to space research that we've seen arise from the programs that we've supported at CIRM. Next slide, please. So beginning with our discovery stage awards, Sonia Schrepfer at UCSF had a project to develop hypoimmunogenic cardiac patches from iPSCs. And so one important hurdle that has always existed with cell therapies is rejection of the graft by the immune system. So we've had several CIRM projects that attempt to engineer cells to evade the immune system, employing different methods to protect the cell therapy from B cells or T cells and the innate immune system. Next slide. So Sonia Schrepfer also has a project aboard that will go to the ISS. She has a project to look at the effect of immunosenescence from CD8 positive T cells on mesenchymal stromal cells as a model for bone healing, and endothelial progenitor cells as a model for vascular regeneration. So microgravity is a model for aging in this case, and the hypothesis is that in microgravity, there will be an increase in terminally differentiated CD8 positive effector memory T cells, and that can impair stem cell-based health in tissues. So these experiments set up broader questions that you may ask in space, including what is the immunogenic profile in microgravity, or what happens to the immune response in space? Next slide. So Jean Loring at the Scripps Institute in Aspen Neuroscience had a later stage discovery award to make an iPSC-derived dopaminergic neuron that's ready for translation. So this is an autologous therapy, and she was doing many of the studies in her CIRM award that were needed to establish reproducibility of the product, including establishing differentiation to dopaminergic neurons for 10 patient lines, determining the functionality of those patient neurons, assessing the immune response of that product, looking at cryopreservation, and then looking at dose response and dopamine release for function in vivo. So a lot of the product attributes that Kelly touched on. Next slide. So in partnership with the New York Stem Cell Foundation and Space Tango, as Janice Studemeyer described, and also the Parkinson's Disease Patient Advocacy Group Summit for Stem Cells, several of the iPSC lines that were created from Parkinson's disease patients are going to the ISS. So neural organoids will be created from these from the patient iPSCs to determine how microgravity affects their migration, neuronal changes, and a gene expression pattern. So deriving mature cells from pluripotent stem cells has long been a challenge in the field. And so a broader question is whether microgravity affects the function of iPSC differentiated cells such as these, or could possibly improve them. Next slide. So Joe Wu at Stanford has been supported by several CIRM awards, including a disease team grant to get embryonic stem cell derived cardiomyocytes scaled up and tested for a clinical trial, and a research award to create a bank of iPSCs from cardiomyopathy donors for disease modeling and drug screening. Next slide. And so his group has built a large iPSC collection of cardiomyopathy samples. And this is a project that the previous speaker, Dr. Arun Sharma, described when he was in Dr. Wu's lab, looking at iPSC-derived cardiomyocytes that were sent to the ISS compared to their effects on normal gravity. And he went through the data much better than I could, but there were some differences between the cells that were sent to space and the ones that were left and regular gravity on Earth, including some hints of possible differences in beatings and arrhythmias. So one question is whether stem cell models like this could predict human health with long-term space travel. For example, could they be used as a screening tool to predict whether a person is at higher risk for heart arrhythmias in space? And is there genetic signature that could be associated with that? Next slide. And so in one final example of a CERN-funded investigator is Dr. Clive Svensson from Cedars-Sinai Medical Center. He's funded by CERN for multiple clinical trials using stem cells. One trial involves using human neural progenitor line that expresses the neurotrophic factor GADNF to treat amyotrophic lateral sclerosis, or ALS. The other trial uses the unmodified neural progenitor lines to treat the eye disease retinitis pigmentosa. Next slide. And so the group plans to send 2 types of cells to the ISS in partnership with Space Tango. The GDNF neural progenitor line used for the ALS trial and undifferentiated iPSCs. And both Janice Studemeyer with Space Tango and Dr. Sharma are probably involved in these projects. So one challenge with PSC manufacturing is to maintain the stability of the cells with passage and expansion. So pluripotent stem cells can lose their pluripotency potential, and they can become karyotypically unstable with prolonged culture and expansion. However, expansion is critical for many PSC-derived therapies to set up working cell banks so that you have enough to go into the patient. And so one question is whether these characteristics can be better maintained in space. Next slide. So these are some examples of CERN investigators that have studied various aspects of stem cell biology in space in connection with their translational and clinical efforts on Earth. And at CERN, we've encountered many other bottlenecks over the years with projects that we've supported. Kelly has touched on a lot of them, but other bottlenecks that we've seen that perhaps space could address are organoids. When organoids are put together, usually they are still in a fairly immature state. And so the question is, if you assemble the organoids in space, can the self-assembly of organoids create more mature populations or different populations than you could see on Earth? Another challenge that we see on Earth is the bottleneck in expanding somatic adult cells in vitro. So hematopoietic stem cells or liver hepatocytes, they either self-renew or they can regenerate in the body. And that's one of their characteristics. Interestingly, when you take them out and you try to grow them in culture, they act like they're quiescent. They really do not expand. And it has been a challenge for decades to get them to do this. The question is, if you put this in a microgravity environment, can you adjust the conditions to allow them to do this? Also, cryopreservation. So cryopreservation is a significant issue with cell manufacturing for your product. You want to cryopreserve the cells to make the cells more feasible for distribution and transplantation, but there is a drawback in that cryopreservation also often leads to a loss of functionality. And so if you can adjust the cryopreservation conditions in microgravity, one question is whether it could overcome this loss of function that you see on Earth. And then the final bottleneck, there are many other bottlenecks, but the final one that I'll just touch on is genomic stability with iPSCs, which we heard about, which I described at the last example with Cedars-Sinai. So this is a significant issue with cell manufacturing, And it'll be interesting to see with this CEDARS project if microgravity can improve that clonal stability. Next slide. So another recurrent issue that we see is that phenotypes can vary significantly among cell lines. So one cell line may respond very strongly to an experimental condition, and another may not have any effect at all. And so with PSC lines, some PSC lines differentiate to certain tissue types better than other lines, and it's really not known why. So looking at cell line variability is very difficult aboard the ISS because, as was described very well by Janice Sudermeyer, there are very tight confines with these experimental cabinets. So in addition to funding research investigators with the examples I described, CIRM has also created resources that exist that assist the entire scientific community, and this is part of our infrastructure pillar of programs. So one of the resources that we've established is an IPS bank. And this IPS bank is currently the world's largest collection of publicly accessible iPSCs with over 2,600 lines. It's also one of the most diverse, with lines represented from African, Hispanic, East Asian, South Asian, and European ancestries. So the cell repository is managed by Fujifilm Cellular Dynamics. And several years ago, CIRM and Fujifilm began a partnership with the Broad Institute in Massachusetts to utilize the breadth and diversity of the collection to create pools of stem cell lines termed cell villages. Next slide. So in the system that the Broad established, up to 100 lines can be pooled in a single culture and later stored for a specific phenotype. So using natural variations in genomic sequences as barcodes, individual cells can be identified that have low to high phenotype characteristics, and a new analysis technique that they developed called census-seq can profile rare alleles that are associated with the phenotype. So the proof of concept experiments for this approach map the effects of several alleles on drug responses for spinal muscle atrophy. And so because this technique enables population scale experiments in very small pools with high internal control, one could see it being utilized eventually aboard the ISS to examine genetic effects of diverse phenotypes in a confined compartment. Next slide. And so this is the final slide that I'll go over. As a funding agency, CIRM wants to support stem cell technologies to advance its mission. And if there are organizations in CIRM that are in California that are developing tools that utilize stem cells for the ISS, the agency has a single funding opportunity this year to support it. As Kelly talked about, Discovery Tool Awards will support tool discovery that will demonstrate technical feasibility in a research prototype. So the tool must use human, stem, or progenitor cells, but otherwise it's fairly open what you can apply for. This award is $250,000 for 12 months. And if you're interested, the deadline for these DIS2 awards applications is August 31st. And more information can be found on CIRM's website, cirm.ca.gov. Next slide. So if there are any questions, please feel free to ask Kelly or me at the information on the slide. Thank you very much for your attention.
Thank you, Stephen and Kelly. That was fantastic. I'm sure the entire community appreciates CIRM's support. And you also did a really, really nice job of kind of encompassing what a lot of the question posers are asking, which is, can you summarize why microgravity may be able to address some of these very basic questions and enable breakthroughs? So if everyone else will please turn on your camera so that we can engage with our audience. We've got a number of questions. We're a little short on time, so if possible, we'll kind of zip through some of these. We won't be able to get to everyone's questions. I apologize for that. There are a lot of them. But I at least I want to remind everyone that this webinar will be available on the CASES website. Probably in a couple of weeks, we'll get the recording there, issnationallab.org. And okay, so first question, I'm going to go back to Dr. Zubair. regarding stem cells in microgravity, could the immunosuppressive response of stem cells be related to adaptation to spaceflight? And second part of that, are there benefits to that immunosuppressive potential that could translate to clinical use here on Earth?
Yeah, a very good question. It's hard to say it's adaptation alone. I think there is more to it because so when the cells return back to our lab, I mean, that's when we assess their immunosuppressive activity. So probably while they are in space, they may not be as much, or so the evaluation is done at Earth's gravity. That means it could be that something they acquire as an adaptation, but clearly that is maintained beyond the microgravity. So, but again, more to be done. Obviously, as you all know, one experiment doesn't cut the chase. So,
well, then let me add a follow-on. If you were to conduct a follow-on experiment, Abba, what kind of an additional experiment would you like to do that would kind of improve on or build upon your results already?
Absolutely. The first one is the question that was raised. Is MSC, mesenchymal stem cell, more immunosuppressive in real time at ISS compared to the ground control? Obviously, we want also to really go deeper into looking at T cell subsets, what T cells are impacted, and do real-time analysis, looking at T regulator T cells, even the cytokine that we assess. Of course, the cytokine are condition media, so it's a snapshot of what is going on in space because the cells were frozen, the condition media was frozen in space. But if you noted, so like CD40 ligand is known to be immunosuppressive, but the level went down. So it's kind of course, the immunosuppressive nature of the that we have ligand or even IL-10, which also went down. So there is more to be done, and we would love to do real-time analysis rather than post-flight analysis.
Okay, one more final question for you, Dr. Zubair. Would you, or have you considered using spheroids in your research? And do you consider that there's a possibility that the results might be different with spheroids?
Absolutely, I think it would be different because with spheroids, you know, it's multicellular environment, and you can there would be interaction of the cells. Obviously, just having cells by themselves, we know in real life it's an ecosystem rather than single. So yeah, absolutely, spheroid is something that we would like to do.
Okay, couple of questions for Dr. Sharma. Arun, did you observe higher levels of dedifferentiation of your human-induced pluripotent stem cells in the space group when compared to ground? And how do those changes that you mentioned in the mitochondrial pathways correlate with maturation of the cardiomyocytes? And third, is it possible to pharmacologically recover the differences? And you're muted, sir. Oh, Arun, we're having trouble hearing you. So if we can't get you, we'll try somebody else.
Okay, can you hear me now?
Yes, sir.
Okay, so I'll see if I can answer some of those questions. Yes, so the question about dedifferentiation, that's actually not something that we observed on the space-flown samples. Of course, there are plenty of other examples of RNA sequencing done on stem cell populations that have shown a more immature phenotype of cells in microgravity. That's actually not something that we observed in our populations, perhaps because these cells are terminally differentiated, so they're months and months old, These are aged cardiomyocytes, but that is in a progenitor population, that would be definitely a more relevant question. In response to a small molecule or something that would be able to mimic some of these effects, yes, I think that is an absolute natural next step. You want to be able to harness some of these biological phenomenon that microgravity might be able to elicit and distill them down to a small molecule format, because that is ultimately something that's very translatable. If you can take some of those observations and some of those discoveries from the microgravity environment and actually replicate them on a ground slide level, I think that would be a tremendous use for commercialization, and a small molecule approach is of course a good way to do that.
Okay, fantastic. Another question for you. How might the changes you saw in microgravity compared to those seen in patients in heart disease or congestive heart failure?
Yeah, so the changes that we're actually seeing in microgravity, and this is not even exclusive to the cardiac side of things. What you see a lot of times in microgravity is an accelerated aging phenotype, right? And that is a lot of those same gene expression changes and functional changes are seen in individuals with congestive heart failure. So there are parallels not only in the functional output of those the functional phenotypes of those two diseases, but also in the gene expression levels as well.
Very good. Let's see, one other question, and I think you alluded to it, but did you find any alterations in the epigenetic regulation of your cells which were cultured in microgravity? Yeah, that's a fantastic question. It's something that we want to address in the future, and I think actually some of the follow-up experiments are going to be focused on that. We actually did not look at epigenetic regulation through like ATAC-seq and histone methylation, those sort of things, but there are a variety of assays that have been developed actually in the last few years that would make this much more accessible now than it was when I actually did this experiment 4 years ago. So that is a reflection on not only the questions that remain to be answered in the microgravity context, but in the technologies that have just been rapidly progressing over the last 5 years in stem cell biology. Indeed. Thank you for that answer and for that look ahead as well. Quick question for Jana. If a researcher is interested in conducting experiments in microgravity, how do they get access to your system, your hardware, to see if there's compatibility?
Sure. Yeah. I mean, we're available obviously through our website, and my email contact, I don't think I put up, but it's at the end of my slides. So, certainly feel free to reach out. And we're very open to talking to all kinds of teams about the work that they want to do. It's so exciting to see the first experiment, the long duration culture that was done by Joe Wu's lab with Dr. Sharma, right? I mean, to me, that was one of the seminal moments when I sort of came across from industry to microgravity, and to watch the changes in the field and what's happening with the capabilities to support the more complex science on orbit. I just think it's a really exciting time to be able to try to answer some of these questions, really accelerate the science, and happy to speak with anybody who's interested to join us in really trying to understand. It's hard enough on the ground to understand what's happening with stem cells, and then adding microgravity as a tool that can help you to understand and maybe unlock some of that mystery of what makes a stem cell and why does it act the way that it acts, is really just an exciting opportunity.
Absolutely. And we want to be respectful of everyone's time. We've actually run out of time. I want to thank our panel members so much. Thank you for your time, your dedication, and your inspirational talks. And with that, I think we will sign off. Sorry we didn't get to any questions for Kelly or Stephen, but I'm sure there's some intrigue and interest with your potential funding opportunity that you might see. So thank you, everyone.
Thank you so much.
Thank you.
Yeah. Bye.